Welcome everybody to the Playing Games podcast. And today in episode 27, we have a very special guest, Dr. Hornberger. Dr. Hornberger, how are you doing today?
Good. How are you?
Good, good. Thank you again for taking time out of your day to join on here. For those of you who may not be familiar with Dr. Hornberger, which if you are reading current research on hypertrophy, I would assume that you are. Why don't we start off, if Dr. Hornberger, you could introduce yourself a little bit, maybe your background and education and what has brought you to study muscle hypertrophy.
Sure. Good question. Fun question to answer. I'm lucky to have a career where I'm basically playing every day. So, how did I know this is what I wanted to play with every day? I didn't know that as an undergrad. As an undergrad, I went to Lake Superior State University, which is in the UP of Michigan, in Sault Ste. Marie, which is also called Sault Ste. Siberia, basically the last frontier of man, going north in the United States of America. So a very cold place, small school. I went there to be a fisheries and wildlife biologist. Quickly learned that I love fishing for sure. I still do, but just that career path wasn't really for me. I really enjoyed the biology, the chemistry. So I became a biology major, and then I was having a really good time at college. Didn't really want to graduate, so I said if I stick around for another year, I can get a chemistry degree as well. And as part of that, getting that degree, I had to do a senior thesis project. And so it could be a project on anything you wanted to study, any kind of experiment that involved basically some kind of chemistry, biochemistry, etc. And I was really into bodybuilding at that time, ergogenic aids. And so I said to myself, well, let's get some of my friends. Half are going to drink a placebo. The other half are going to drink a ton of L-arginine, which was the hot supplement at the time. And I had this idea that L-arginine combined with intense exercise, basically anything that would get you sort of past the lactate threshold, would cause growth hormone secretion. And so I got my friends, placebo versus L-arginine drink. They're on the bike at literally the public gym at Lake Superior State University. I had no approval to do human work, no IRB approval or anything. I had a friend who was a major in nursing. They got all the blood draw equipment. So I'm giving them the drinks, pushing them on the bikes. My friend's taking blood draws just in the regular gym. And somebody from the exercise physiology department came by and saw me taking blood from people and was like, what are you doing? And so I explained that. I was like, this is my senior thesis project. I got this great idea. I'm going to measure growth hormone secretion. And he said, what you're doing is called exercise physiology. And I was like, well, what is that? And he's like, well, there's actually like a field of scientists that actually do this for a living. And I was like, oh, okay. And so he said, you should come to this ECSM meeting that we're all going to in a month. And you'll see, a lot of research, just like what you're doing for fun in the gym. And I was like, great. So went to ECSM meeting, and I kind of immediately knew like, oh, I could do this for sure for a career. Like this wouldn't ever be work. So after that meeting, it's like I need to go to grad school, and I was really interested in ergogenic aids at the time. So I went to UT Austin. I worked with John Ivey initially, who's world renowned in his work in ergogenic aids, and worked with John Ivey for a little bit. Ended up sticking with Roger Farah. Both labs, I felt like the work was really like very applied. I wasn't able to utilize sort of my knowledge in chemistry and biology the way I wanted to. And so I ended up just getting a master's at UT Austin. And then I went to University of Illinois, Chicago to do my PhD with Karen Esser. And there I really got into the molecular biology and became interested and focused on like, what is it that happens when you apply mechanical load to muscle that drives growth? What is that signaling cascade? What are the mechanosensors? I wanted to know everything about that process. And that hasn't changed. It's still the same question today. So here we are like 25 years later, and I'm still playing around with the same questions. So I like to think that we answered a few things along the way, but the passion and the interest is not wavered at all. So
when you did your PhD with Dr. Esser, how was that experience? Because I know she's probably one of the most respected researchers in the field, and is very she's very intelligent or very precise when she's doing experiments, and
Yeah.
she's done a lot of great work. So I don't know if you have any great experiences with her. She
Oh
taught you a
yeah.
lot.
So I refer to Karen as my academic mother, because she really did raise me, to be an academic. I came, like, as I mentioned, I came from a small university in the middle of nowhere in this UP of Michigan. Got more formally educated at UT Austin, but in terms of really being trained rigorously to think critically, to always be answering that question of like, so what? What you made this observation. Why should I care? So what? She would ask me this question every day. And so she really, pushed me, to become a better scientist and to think critically and to think about like why what I'm doing is actually really important. And every aspect of any success I've had, I attribute to the training I got under Karen. So yeah.
That's awesome. Yeah, I think it was a very fortunate time for you to become a PhD with Dr. Esther, because I don't think she no longer does this field precisely.
Right.
Uh,
Yeah, she changed after I left. I mean, at the time, I went to Karen, because Keith Barr, who you're probably familiar with, had his work on P70S6K phosphorylation following it was the seminal paper that he published back in 1999, and showing this correlation between the activation of P70 and protein synthesis. And I was like, that's what I want to do. Like, I want to study like the molecular mechanisms that drive this growth response. And it was that paper that made me go like, okay, I need to contact Karen. I need to work with Karen. I got a funny story about how she decided to take me. If you want to hear a funny story.
sure, yeah.
Okay, um, so I've always been really good in math, quantitative. So back in the days, we had the GRE, and it was like a 1,600 point scale, and you had three categories. You had like it was verbal, quantitative, and analytical. And quantitative and analytical, I was very, very good at. I never had to really work at that at all. Um, Verbal, not so good, or written skills, not so good. But I needed to get like a 1,200 to get into the grad program. So I went and took a couple practice tests, and I scored like nearly perfectly on the quantitative and analytical. And so I was like, all right, whatever I get on the written, like I'm going to get the score I need. And so I took the formal test, and I got 16th percentile on the verbal and written part. So Karen calls me. She's like, your application looks great. Your letters of reference look great. The reason you want to come to my lab sounds perfect, but I needed to talk with you on the phone to make sure that you can even speak English. She's like, how do you get 16th percentile? And and I was like, I don't know. And she's like, writing's a really important, skill in this program. And I'm like, absolutely. I was like, read my thesis, read the paper that I'm about to publish. I was like, I think I can write pretty well. I don't know. I tanked the exam somehow. And so she ended up accepting me, obviously. And I've written a lot of papers since, and I like to think that they're at least fairly legible and not worthy of a 16th percentile score in the GRE. So the long story there is like, don't let a big exam like that and a poor score define who you are. I don't think it really necessarily does.
Absolutely. No, I think that's a good message at the end there. That way, or that said, to get started on the questions, let's start off with kind of the foundation where we're gonna begin talking off of. So, which is, how do muscles grow, or in what ways can muscles grow?
Yeah. Which level do you want to talk at? Whole muscle level, fiber
Let's talk
level?
about let's go maybe from the muscle and then probably work our
Sure.
way down into the fiber
Okay.
level.
Yeah, start at the whole muscle level. I mean, in humans, the most predominant mechanism is going to be radial growth, and so an increase in the cross-sectional area of the muscle. Once you're an adult, longitudinal growth, increase in the length of the muscles clearly happen during development, but once you're an adult, there's not much change in muscle length. You can do some certain extreme things like limb lengthening and stuff like that to get the actual muscle length to increase. But there's not really much in terms of longitudinal growth at the whole muscle level once you're an adult. It's all radial growth. Yeah. It doesn't mean that's true in a level of the fibers or the myofibrils, as we'll get into. Yeah.
Yeah. So how would that change at the level of the fibers or at the sarcomeres?
Yeah, I think there's very compelling evidence that at the level of the muscle fibers, the fibers can grow in length, and that seems like counterintuitive to say, well, the muscle can't, but the fibers can. But what you have to appreciate is that the fibers don't run usually the full length of the muscle; they're at a pennation angle, and so you can have these fibers grow in length without the actual length of the muscle changing. And at least all of our mathematical modeling suggests that that's an important component of the growth response. For sure, in more extreme models in mice, like chronic mechanical overload, but there's also very good evidence, I think, in humans that longitudinal growth happens. So you can see it at the level of the muscle fascicles in many, many studies. Yeah.
So getting into like a little deeper on this, when we talk about radial growth, we mean a lot of people just mention sarcomeres in parallel, and then when we talk about longitudinal growth, we just mention sarcomeres in series. But
Yeah.
when we think sarcomeres in parallel, I think some people get confused with like how do sarcomeres just like add each other on top of each other? Like is that hyperplasia, or how do sarcomeres like create formations to start new sarcomeres? Do they create new Z lines, something like that? I don't know if you want to talk about a little bit more about the mechanisms.
Let's see. So I'm completely winging a visual example here. um, okay, so those are myofibrils, right? Um, and so you could make your muscle fiber bigger by adding more myofibrils, right? Um, you could also, and this is where you talk about adding sarcomeres in parallel. So adding those more myofibrils would be adding sarcomeres in parallel, but you can also make your sarcomeres bigger. And so you can get radial growth without the in parallel addition of sarcomeres. And I think that the example that you gave is not really entirely comprehensive. Does that make sense?
Yeah.
Yes. Okay. Okay. Um, so I kind of lost track of the question. What was the question
Uh, Mechanistically, at least for sarcomeres in series, how do muscles kind of add sarcomeres in series? Like, do they break down
again?
their Z line, create a new one? And then for the radial growth, when you mentioned the sarcomeres can get bigger rather than being added, um, is there a mechanism that happens first? Like, do they get bigger first and then they add each other, or is it vice versa?
All questions you're trying to answer. I'll deal with the in series first. We just published a paper on this. I imagine you're aware of it. That so our data suggests, it's definitely not definitive, but I think it's pretty compelling that you can get the addition of new sarcomeres all along the length of the myofibrils or the muscle fibers. So there's this dogma that used to be that you only add new in-series sarcomeres at the ends of the fibers. And we don't see any evidence, compelling evidence to support that. Instead, our studies strongly suggest that new sarcomeres can be added throughout the length of the fibers. And we see evidence of this occurring at regions that we call transverse splitting. So imagine like here's a sarcomere. If this is a Z line, what we think happens is that Z line breaks apart and you start to form a new sarcomere in the center of that. And so we've done recent studies using a technique called BondCat, which labels newly synthesized proteins, but basically can label like where new sarcomeres have been formed. And our data nicely aligns with a model in which individual sarcomeres break at that Z line and then you form new sarcomeres in the middle. And this can happen all along the length of the myofibrils.
Not just at the terminal ends of the muscle,
I,
right?
I won't bash the original author of who came up with that idea and created that dogma, but the dogma is very heavily ingrained in the literature, but there's very little evidence to support it. Yeah,
Interesting.
yeah,
And then
that's
as
my
for
opinion. My opinion. So
radial growth, you said the sarcomeres can get larger themselves. Do they
yeah,
do that before they add on each other, or do they add on each other and then they get bigger? I think
sure.
the
So
previous one was.
the same author who had the idea about new sarcomers being added at the ends. So this is Goldspink stuff. He had lots of great, beautiful ideas. It just not necessarily all of them ended up correct. Is kind of what I'm trying to say. So I don't want to sit here and bash Goldspink for having really brilliant and novel ideas, which I believe that they were. So Goldspink's ideas were that you would first expand the size of the sarcomere and reach a critical size where then it would begin to split and you'd form two new ones. So he called it myofibril splitting. And there's a little bit of evidence in the literature that you get an increase in myofibril splitting or signs of it as myofibrils get bigger in size. And so our thinking was, okay, we coined this concept called the myofibril expansion cycle, in which we thought, okay, in response to increased mechanical loading, the first thing is the sarcomeres get bigger in size and cross-sectional area. Then they'll split, they'll form new daughter myofibrils, and then those ones can grow in size and split and so on. And so that sort of became what we thought of as like this myofibril expansion cycle. So we've done a lot of studies. Many of this has not been published yet to try and interrogate the validity of that idea. And the first thing we see is like in time, this is all in the mouse chronic mechanical overload model, but we see similarities, although not as exhaustive in our data in humans. But in mouse, it's pretty clear that there's this rapid increase in the size, the cross-sectional area of the sarcomeres, and then that sort of plateaus. And then over time, it starts to come down, which is in line with what you'd think of in the myofibril expansion cycle. The first thing that happens, they get big, and then they start splitting in size and go down. Again, consistent with that myofibril expansion cycle is that the number of myofibrils per fiber doesn't change at all for the first many days, and then starts to come up. So it's sort of this later response, whereas the increase in myofibril CSA is really fast. Myofibril number comes on later. So all the kinetics of that align with the idea of the myofibril expansion cycle. However, when we've gone to try and like look and see if there's actually signs, increased signs of splitting, we don't see hardly anything. Like it goes from like 8% of your myofibrils have signs of splitting to 8.5. Maybe statistically significant because we measure with an automated program literally like millions of myofibrils. But that magnitude of change just doesn't resonate with the magnitude of change in the number of myofibrils that we see. And so the other possibility of how you get new myofibrils is it's called de novo myofibrogenesis. So you can form completely new myofibrils. And we have one published data that at least shows signatures that that's probably the primary way in which you get an increase in the number of myofibrils per fiber. So
interesting. I didn't hear that last one, but it does sound more plausible in my head than just the idea of sarcomeres being added in parallel. I feel like that leaves out or leaves more questions than answers. Now the thing is, a lot of I think most people are familiar that if you increase radial size, you will increase muscle cross-sectional area, as you said. But you've published a letter to the editor, and I think you've also worked on a few other papers where you mentioned this, that as you increase the length of the sarcomeres, you can possibly also influence mid-belly cross-sectional area.
Well, I would say as you increase the length of the fibers.
Fibers.
Okay, yeah. Correct.
So how could that happen? Maybe if you could explain what you guys wrote in that letter to the editor, maybe kind of simplify that mathematical model, because some people might look at that and still be left a little bit confused.
Okay, so you wanted me to explain to you how an increase in the length of fibers could increase the cross-sectional area of the muscle. And so what I have in here is a hypothetical muscle that's modeled based on the mouse plantaris. So like everything is exactly to scale of what you would find if you were to look at the individual fibers in a mouse plantaris muscle. The pennation angle is correct, the diameter is correct, the length of the muscle, everything is exactly to scale. And if we zoom in on here, what I'm showing is in gray is the length of the original muscle fibers. And then in red the length of the fibers. I think if in this example we increased it 13%. Okay, so what I've done is every single fiber I increased the length 13% throughout here. Notice that the length of the muscle doesn't change one bit, right? If we took a cross section through the middle in gray, that's the cross sectional area of the original muscle. That would be the in pink, the new cross sectional area.
Okay.
And this kind of highlights sort of a different perspective. So if you were to imagine like taking a cross section of this muscle and you counted the number of fibers in a cross section that went through that mid belly cross section, you have in gray that shows 48.4 go through that individual cross section. After the longitudinal growth, there's 59.7. So more fibers are making their way through the cross section just because of the geometric or architectural properties of the muscle itself. Again, muscle length didn't change, fiber length got bigger, muscle cross-sectional area got bigger, and that was entirely driven by an increase in the number of muscle fibers per cross-section. And that increase in fibers per cross-section was not hyperplasia. It was from longitudinal growth. Yeah.
So, and that's a really interesting model. And it led a lot of people to start suggesting that whenever we perform, let's say, eccentric contractions versus concentric contractions, a lot of people would then go on to say that main growth from eccentric contractions is sarcomeres in series, and it's those sarcomeres that are being added in series from eccentrics that cause the increase in cross-sectional area similar to that of concentric contractions.
Yeah. Do you want me to stop sharing?
If we're no longer going to use this, yeah.
Yeah, I think it'd be fine. I know how to pull it up quickly now if we need to do it again. So, okay. So yeah, I don't know the intricacies of every study that's been published on eccentric versus concentric growth. I think my sense is that eccentric versus concentric exercise and growth. My sense is that there is a general trend if you're doing eccentric, everything else being equated, you're probably going to get a little bit more growth in terms of just whole muscle than you would if it was purely concentric or isometric exercise. Do you agree with that take on the literature? Does that sound right?
Yeah, when we're doing eccentric overloading, right? When we match, right? Yeah.
Everything else match, right? And so I would lean toward into the argument that that extra growth is probably due to augmented longitudinal growth. Perhaps there's an augmented cross-sectional growth, but studies indicate that like eccentric exercise is the most potent for inducing longitudinal growth of fascicles and presumably longitudinal growth of the fibers. So my thinking is that that augmented growth that you might see with eccentric based exercise is because you probably squeeze out a little bit more longitudinal growth in series addition of sarcomeres. Yeah.
Mm-hmm. Yeah, I agree with that. I think that makes sense. And so on top of eccentrics, another important thing is loading in a more stretched position. A lot of people think that if you stretch for a long time, or if you, like, let's say you're doing an exercise and it's heaviest where the muscle is most stretched, people think that that could signal sarcomeres to be added in series. And therefore that extra growth from the stretch could be due to those sarcomeres being added in series, influencing the cross-sectional area. However, there is some debate about that, saying that the number of sarcomeres that we add, I think this was from Herzog, that we add in series with, like, from what we've seen, is very, very minimal. And so he suggested that it may not be enough to influence cross-sectional area if we don't add enough sarcomeres in series.
So the caveat here with any research that's done in humans and making conclusions is that those measurements of the in-series sarcomeres per fiber are based on measurements at a few isolated, spots along the length of the fibers. You can't take whole muscle fibers out and actually count the number of in-series sarcomeres. So it's based on extrapolations that may or may not be very accurate. And there's very good reason. If I just trained the crap out of my calves a week ago, and I know that parts of my calves, my gastroc got absolutely obliterated. The distal part of my gastrocs were obliterated. The proximal part wasn't. I can still feel it today. It's 5 days later. So the things that happened at the distal part are definitely going to be different than what happened at the proximal part. And so if you didn't look in the right region, you miss it, right? And so by like taking measurements at just a few discrete spots along the length of the muscle or a single spot, which is usually what happens, I don't think you can make very compelling conclusions about what might have happened along the entire length of the fibers. That heterogeneity in the growth response within the muscle belly is something that's often ignored. But I'm guessing you go to the gym and you train, you probably felt this before too. It's not like the
Yeah.
whole muscle belly. Sometimes the whole muscle belly hurts, but sometimes you do an exercise in a certain way and it's only part of it.
Yeah.
Oh,
And well, I was gonna follow with sarcomeres again. Okay, yeah, that makes sense. So if you have regional, because muscles, there's regional hypertrophy within muscles, and
yeah.
at least transiently, I think we see that. I don't know how it looks long term. I don't know if anyone has done that, but
Yeah.
transiently we do see growth more in one part of the muscle with the other. So it could be that maybe that growth could have been with sarcomeres, but the measurement was in the other side, so we probably didn't see it. Right? Okay, yeah, that makes sense.
And there's actually a decent number of studies that have looked at regional differences in growth along the length of muscle fibers. Some are actually like MRI studies that have gone the entire length of the muscle itself.
So regarding the model about sarcomeres increasing cross-sectional area, I assume that doesn't apply to every single muscle equally
Sure.
because muscles have different architecture types, and I mean, there's like what, 8 different architecture types that I'm aware of. I don't even know how it applies to all of them. But one of the popular ones, let's say, is the biceps. The biceps are very fusiform, which means that the fibers, if you look images of it, they run down kind of like almost straight down to the origin from the insertion from the origin to the insertion. And so I think this goes back to an equation you've had in one of your papers where you talked a little bit more about this model where there are limitations to it. So let's say, I don't know if I remember the equation uh, correctly, but it was something like fibers over fascicles times the cosine of theta. And depending on that, um, you could add sarcomeres in series and it won't influence the cross-sectional area of the muscle. I don't know if you want to kind of elaborate or build up on that.
So effectively, the equation that you mentioned defines whether or not a muscle will have all of its fibers crossing through the mid belly. So if you think about the example we gave, the cross-sectional area got bigger because the fibers got longer, and that caused more fibers to go through the mid belly. Do you remember? Okay, so if you have a muscle where not all the fibers go through the mid belly, then an increase in fiber length can have more muscle fibers working their way through the mid belly and allowing an increase in fiber length to contribute to an increase in fiber cross-sectional area. In the sort of extreme opposite that you mentioned in the bicep, where the fibers generally run the entire length of the muscle, you have very little pennation angle. Every single fiber goes through the mid belly, right? So you couldn't get an increase in the number of fibers going through the mid belly from longitudinal growth. You wouldn't get that increase in muscle cross sectional area through that mechanism.
Okay, yeah, that makes sense.
Okay.
And do you think there's a limit unto how many sarcomeres you could add? I'm sure there is, but in series. But I think when we build muscle, I think there could be a slight change in pennation angle. Like, I mean, the biceps, I don't know if they can change like some sort of pennation to pack in more fibers as they get a lot bigger. Maybe they can. I mean, even muscles like those, they have like a teardrop end where they kind of
Sure.
curve. So maybe if you increase muscle, that could increase some of the capacity to continue adding sarcomeres in series. Do you think that's a possibility?
Imagine like you've got this is the outer edge of your bicep, right? And the fiber runs the full length. It's got to be a set length, okay? And now if that muscle gets bigger, that fiber has to run a longer length to go from end to end to get around that full circumference,
Hmm.
right? So even there is an example where longitudinal growth would be important. It gets really complex because the geometry of all of this is it sort of becomes um, like what do you call it, four-dimensional chess in a way. You really got to think about a lot of nuances in it. But
Are you planning to publish more work in that specific topic in the future?
on longitudinal growth, or
Or like how it could impact muscle hypertrophy, maybe,
well,
or cross-sectional area?
in terms of modeling, not so much in terms of modeling. Like we've really been interested in just like what are the mechanisms that make sarcomeres have a bigger cross-sectional area or myofibrils have a bigger cross-sectional area. What are the mechanisms that make myofibrils longer? The in-series addition of sarcomeres. What creates brand new myofibrils? What are the mechanisms of de novo myofibrogenesis? Those are sort of the core of where our interests in the lab are at. We like to really drill in deep and get down to very minute questions.
So, I mean, I think this is a good time now to finally transition into the signaling pathways of muscle hypertrophy. Your primary interest, I think, right?
Yeah.
So, when it comes, I know you published, I think, two recent big papers, like huge papers. I think one of them was huge, um, with the signaling pathways for hypertrophy. What goes on? Like, where do you even start with that? What do you guys find are the main signaling pathways? How has that changed from the past? Are there any that still kind of are holding up, and have there been any that we no longer kind of look at?
Uh, So I mentioned I went to Karen Esser's lab. I was inspired really by Keith Barr's paper in 1999, which kind of was the first paper to suggest that mTOR might play a role in mechanically induced growth. And so I sort of hung on to the coattails of Keith's work and really became interested in how mechanical stimuli activate mTOR signaling. And that really became the crux of my career for 15 plus years. Um, and our early thinking and all the data from the sort of the beginning, if you go back 20 years ago, was like mTOR is the be all end all. It blocks, if you don't have signaling through mTOR, you don't get radial growth of muscle fibers. It blocks the increase in protein synthesis. So it's like everything is like all about activating mTOR. But in the last 10 plus years, it's become pretty clear that mTOR is part of the equation, but not all of it for sure. So, for instance, in the increase in protein synthesis that occurs, there's numerous studies now that show that you can get an increase in protein synthesis through a completely mTOR1 independent mechanism in response to resistance exercise, in response to chronic mechanical overload, even in response to aerobic exercise. So lots of examples out there that mTOR is not explicitly required all the time for every given situation. And so one interest we've become focused on is like, what is this mTOR1 independent pathway that drives an increase in protein synthesis? And then still ultimately, what are the mechanical sensors that initiate the whole process to begin with? That's where I began. That was the question that I was passionate about answering, and that's the question I'm sure I'll go to my grave without knowing the full answer to. But I'm hoping to make continuous headway towards getting closer to answering that question. And so I think the paper that you're referring to is the one that showed that MKK3B and MKK6, the prolonged
Yeah.
activation of signaling through those kinases are potentially critical drivers of the mechanically induced growth response. We definitely continue to think that. Our sort of current thinking is that MKK3 and 6 serve as sort of a node, like imagine like an hourglass signaling cascade. Okay. And MKK3 and 6 are two kinases, but they're capable of regulating the phosphorylation of literally thousands and thousands of different substrates. in part because or in large part because they activate P38, and then P38 is a very promiscuous, or multiple isoforms of P38 are promiscuous kinases that phosphorylate all sorts of other substrates. So we envision like MKK3 and 6 as sort of at the top of this little node. And MKK3 and 6, there's at least 20 different kinases that can activate those two guys. So you have this sort of hourglass shaped possibility, like a lot of kinases that can filter into MKK3B and 6. And then from those, you can have this huge plethora of signaling events, which is what we see when we look at the whole phosphoproteome. And so one major question now is, well, one is this concept that they're a critical node. Is that valid? So that's one thing that we're focused on. And then also, how do MKK3 and 6 get activated? We know that they get robustly activated, but what are the kinases that drive that activation? Not an easy question because there's at least 20 known possibilities. So kind of working through those in a rigorous manner is not a trivial task. And we're proposing experiments to try to figure out what they are. I think it's up and leave it at that.
So I know there's a few popular ones. For example, there's focal adhesion kinase, calcium, and IGF-1.
Okay.
Have those views changed over the years? Has one of them become probably less important or less relevant than the others? Is one of them still kind of being worth looked at, maybe like calcium or something?
Yeah. So I'll just tell you my own personal opinions about the roles of those two different potential players, and it's informed by the studies that we've done. The literature is out there on them, and we were, of course, very interested in the potential roles that they play. When I started working on mTOR signaling, I was under the hypothesis, I had a grant funded under the hypothesis that mechanical stimuli would release autocrine paracrine factors such as IGF-1 that would then come back, activate things like the IGF-1 receptor, and through a PI3 kinase dependent mechanism, activate mTOR, and then mTOR would drive skeletal muscle growth. That was the general hypothesis for the grant that I was pursuing. And we tested that, and using a PI3 kinase inhibitor, we found out that actually, contrary to all the sort of momentum at the time, signaling through PI3 kinase was not necessary for the mechanical activation of mTOR. IGF activates mTOR through a PI3 kinase dependent mechanism. So I was like, okay, IGF is not involved, at least in the activation of mTOR. And that sort of excluded all other receptor tyrosine kinase based growth factors. And we also did experiments. If you go back and look at our 2004 Biochem journal paper, I think it's in that paper, we had muscles in a bath immediately adjacent to one another. One was being stretched, the other is just sitting there. And the idea, like, that stretched muscle would release these autocrine paracrine factors. And so we should be able to see activation of the same signaling events in the muscle that's sitting there, in a controlled condition when it's right next to the stretched one. Never got any evidence of that. Did the same thing with like conditioned media experiments in a cell culture bath. So taking cells, stretching them, taking the media off of those cells, putting it on control cells, looking for the activation of the signaling events. Again, no evidence. So as much as we tried to find evidence for a role of autocrine paracrine growth factors such as IGF-1, all the data told us that that's not happening to any meaningful degree that we could detect. So I don't really, I don't think that autocrine paracrine factors such as IGF-1, or at least in terms of the mechanical activation of the signaling events that we see, I don't think it's important. Calcium is another one you brought up. Calcium is, again, something we looked at. And calcium has been implicated. There's a lot of different potential mechanisms through which calcium could activate things like mTOR. We used a pretty robust sledgehammer approach to address this. So a intracellular calcium chelator called BAPTA-AM. So you can put that on tissues. It's cell permeable. It'll chelate any free calcium inside the tissues. We could, as a positive control that it was blocking the release of intracellular calcium, we could use a drug called, I don't remember which one we use offhand, but a calcium ionophore that basically causes the release of calcium from the intracellular stores. Doing that would activate things like P38. When BAPTA-AM was there, it would block it, but BAPTA-AM did not block any of the stretch-induced signaling events that we studied. So that showed us that like changes in intracellular calcium are not driving the activation of things like mTOR. And to me, like the potential for calcium to work it just doesn't make sense in muscle because you're talking about a tissue that just gets absolutely flooded with calcium every time it contracts. And that's true if it's endurance exercise, which doesn't really induce growth, and it's or all these signaling events that we see that happen with resistance exercise. Calcium gets released with resistance exercise. So like, to me, it just doesn't add up. That calcium would make for a good sensor of a resistance exercise specific stimulus. It doesn't mean that it doesn't. It doesn't mean that I'm right by any means. I just struggle to find a way in which one could imagine that calcium would work that way. Yeah.
Maybe I know there's some that say that like it's still kind of like up to debate because it has like some relationship with calcirin, something like that.
Calcineurin. Yeah. Okay.
And then
Um,
they say, well, there's like that relationship, and then that can relate to the hippo pathway, and then maybe that can possibly have some mechanism. Do you think?
yeah, I'll just lean into like I don't I'm open to anything that's correct, and I just want to know the truth, and I think about this all the time, and I haven't seen anything that makes me really think that calcium plays a role in this process. I have seen things that show me that it doesn't. So that's kind of where my mind goes to on this. Yeah,
And I mean, I mean, you could also build muscle with like with active tension where you're contracting it, but you could also build it passively, and I don't
that's
think
right.
there's any calcium in that, right?
Well, I mean, not calcium that would drive the contractions, but there could be very local calcium release to activate intracellular signaling cascades. And this is we were doing passive stretch when we were doing those BAPTA AM experiments. So that was just stretch, like not active contractions. It was just passive stretch. It did not block the activation of P38, of mTOR, of any of these things that we always see. So yeah.
So I guess to finish off with mTOR, it's it's important for muscle, but it's also not needed to build muscle, right? Do you think
Well,
it's important?
no, being needed, being necessary is a different thing than being sufficient to drive a process. I,
Mm.
I think a muscle needs calcium. I'm not saying that. Muscle definitely needs calcium to function and to contract and to do anything really. But I don't see it as being necessary for mechanically induced growth. I didn't say that right. As being sufficient to drive mechanically induced growth, like changes in calcium driving it.
At least not
That's,
in isolation, right?
yeah.
Okay. And then I mean, with mTOR being like the big one that everybody would say it's like the main driver, it seems to be more important with radial growth than it is with longitudinal growth, right? Because muscle can. Do you think it's mTOR is still needed or very important for radial growth, and then it's the one with longitudinal growth where it's not needed, or like it can be
Yeah.
blocked?
So this is really puzzling because so many times now we've repeated, and some of this is still unpublished. A lot of it's published. Um, every which way we bang it, mTOR is necessary for the mechanically induced radial growth of muscle fibers. Every time, we have not found an exception to that. Longitudinal growth, we have published one large study and have multiple other examples that longitudinal growth does not require signaling through mTOR. So there's definitely two different types of growth. They are dependent, varying their dependency on mTOR. The part with radial growth is confusing because Although we can use like a drug like rapamycin to block radial growth in that exact same setting, that rapamycin does not prevent the increase in rates of protein synthesis. So the question is like, well, where do all the newly synthesized proteins go? Um, and maybe part of them are going to longitudinal growth, but it's not all. It can't be all of it. So that's one part that's really puzzling to us. And a Science Advances paper, if you get into the supplemental figures, I think it's like supplemental figure 5. We just published this a few months ago. We propose a rather complex explanation of actually how that could happen. That might be a topic for a different different day. I don't know.
I mean, I would I think we finally came to an end of the discussion, but I will link that big paper down in the comment below for anybody who wants to read it. I know a lot of our listeners are actual students in the field. I did want to ask, do you have any projects in the future where you might look at signaling pathways between different contraction types? Is that a possibility, or is that something that's of interest, or
So,
is it the same?
so you're talking like concentric versus eccentric?
Yeah,
Yeah.
maybe.
It all depends on what the underlying question for that is. We use this passive stretch model. To me, this is for everything that we see in the passive stretch model. That's something I developed back in my PhD. Everything we see from a signaling perspective in that passive stretch model recapitulates what we see with eccentric contractions in the same model, and it also recapitulates what you see with intense contractions in skeletal muscle in vivo. But the passive stretch model is, to my opinion, excellent because it eliminates all the metabolic aspects. So it's a purely mechanical model. It separates out, things get much more complicated when it's metabolism plus mechanical. Um, and yeah, definitely looking at like passive stretch versus any type of active contraction is an obvious thing because that allows us to sort out like this could be metabolic versus this is mechanical. But then getting into the nuance of like concentric versus eccentric. I mean, it's not. I don't have any specific studies that we're doing on it, but if there was a reasonable desire or reason to look at that specific question, we would. Yeah.
Mhm. Awesome. Well, Dr. Hornberger, thank you again for your time. I think a lot of our viewers would have found this call very valuable and probably have answered a lot of their questions. I mean, there's a lot of questions that are still unanswered, but I think with your work, it seems like we're getting closer to somewhere or having a better understanding. I think that's really nice to see because with signaling, it's always a mess. All right. For everybody, thank you for listening again on the Playing Games podcast, and we'll see you on the next episode.